There have been a lot of statements recently or over the years that mRNA can't change your DNA because it's RNA. Now, in order to understand how the cell works, you have DNA, which is the blueprint. The DNA will then make RNA, and the RNA will then make proteins. And so the argument has always been that mRNA can't become DNA. Now, that's partially true because some viruses are able to integrate DNA from RNA, so that's a different point. But in the context of these jabs, the argument has been that there is no concern because it can't alter the way how your DNA works. I think that this recent paper definitely changes that argument. And this is what I'll be talking about today. It's a complex topic, and it's a paper that was published on the 25th of March, Persistent Epigenetic Memory of SARS-CoV-2 mRNA Vaccination in Monocyte-Derived Macrophages. This is an important topic, and in truth, this is being positioned as a wonderful thing. They're saying that, wow, look, this is one of the ways that mRNA vaccines work to train your immune system. Now, the reason they're having to come up with that is because there were recent papers in the past year that demonstrate that mRNA does not produce long lived plasma cells in the bone marrow, like what you would expect with a normal vaccine. And that's a problem because if you're not getting long lived memory, how then can the immune system remember when it faces a virus again or an infection? That was a problem. And so, in their search, it seems, they found something else here that they say, whoa, 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 look at this, look over here. It trains your macrophages to be able to remember and respond to the infection. As I said, on the surface, that may seem like a good thing, but it immediately played red flags for me because I knew instinctively there was something very important in this paper, but not necessarily in the angle that they were looking at. Now, you have to bear with me because this is complex stuff. This is definitely outside my expertise in the sense that this is not something that I study, but I can apply some of my understanding of biochemistry and genetics to try and explain some of these complex ideas. So you have to work with me, and I'll try and see if I can break it down simply. So this is a work in process, but it gives you an idea as to what it is that I'm looking at here. So let me show you this here to give you the basics as to where we're starting. This is an example of how your DNA is structured. And at the bottom here is the chromosome. You have 23 pairs in the body. And this is about 1400 nanometers. And then looped, they're all looped chromatin. It's smaller. This is the smaller bit of this section. This is what it looks like. And then if you take this small section here, this is what it looks like, where you have the chromatin fibers. These are wrapped around. And if you were to look even more closely here, you would see that they are then wrapped. So the strands of DNA are wrapped around like a yo-yo, these circular things. Now, this is what you have to remember. These circular things here, when we open it up further, represent histones. And the strand of DNA is wrapped around it, looped around it, then it goes, loops around it again, and it's wrapped tightly. And this here is what the strand of DNA, double-stranded DNA, looks like. And this has all the instructions to make everything in your body. And so, looking back again from the opposite direction, the long string of DNA, it's then wrapped like a yo-yo around a histosome, which makes a nucleosome. And then that goes on to the next one and the next one. And then they're all bunched together. Then they make these chromatin fibers, which are then looped together. And finally, they become a chromosome. So this is one huge piece of information. And that's really how it works in terms of the cell. Now, what then happens is that you may then be thinking, okay, well, that's the DNA. The mRNA didn't change the DNA, but you just need to understand a little bit more about what happens with the yo-yo. That's the bit that I want you to focus on. And so when we think of these histones, this is what they would look like. And you can see there four globules together with four tails. And in effect, this part of the histone, H3, and its tail seems as though it gets modified by the mRNA, which then keeps this histone in an active state, and critically, it unloops the DNA. Okay, now this is the important bit that you have to get. I'll try and see if I can find a way that you can understand what that is like. Let's see here. Okay, so remember what I said about the chromatin being condensed. So when it's all wrapped up tightly, the DNA, it can't make new RNA. In order for it to make new RNA, you have to unwind the yo-yo. Make sense? So if you imagine the DNA as a string wrapped around a yo-yo, there's a section in the string that you want to get to. In order to get to it, you have to unwind the yo-yo, the string, so that you can get to that section. That's where the histones come in. And this is what it would look like. This is closed or condensed chromatin. And you can see here all of these histones with the DNA wrapped around it. These are like yo-yos. So four yo-yos together, you can't get at the DNA. But when you have this process happening, acetylation, you then have an opening of the yo-yo. And then you can have here an RNA transcriptase, which then converts a string of DNA into RNA, which it can make protein. So this is an important part of epigenetics, because as you remember, if all the DNA is closed up, it can't make protein. The sections that can make protein have to open up so that it can be transcribed. It's this process here that is affected by RNA. So it hasn't changed the DNA, but it has changed the accessibility of that DNA to be written. So a very important differentiation. It's what they call epigenetics. So the DNA is not changed, but in effect, it has changed the ability of a section of DNA to be transcribed. And if you leave it open, it will continue to produce the protein that is in that section. That's the epigenetic change. And so in reality, when you looked at the paper, this is something that they were very excited about, because they thought, wow, great. We have a way to put it forward that the mRNA works to protect against further infection. Sadly, I don't look at it that way. And I'm just looking at it from a different perspective, maybe. But the point being is that it's still relevant. Here is what they were saying. How precisely mRNA vaccines train innate immune cells to shape protective host defense mechanisms were relatively unknown. So just think about that a minute. They have acknowledged that they don't quite know how this works. That's not a good sign to me. And so, therefore, what they're trying to figure out really is, well, why didn't it work the way we thought it should work, like another, say, measles vaccine that trains long-term immunity plasma cells to produce antibodies, T cells to target the virus when it comes back? This seems to be working in a different way. They found that 2 consecutive vaccinations were required for the persistence of H3K27AC, which matched with pro-inflammatory innate immune-associated transcriptional changes and antigen-mediated cytokine secretion. So all of this is happening in a specific kind of immune cell called a macrophage. And essentially what it's doing is that it's locking it in an activated state for at least 6 months. Now, when I think back to some of the strange things that occurred in the pandemic, one of the strange ones was a condition they called hypermetabolic lymphadenopathy. Okay. What they found, and they've done this study in Israel, is that within about 3 months of someone having a COVID vaccine or the mRNA vaccine, specifically for Israel at the time, you couldn't do a PET scan to look for cancer because it would get confusing. They wouldn't know if the lymph nodes were lighting up because of the vaccine or because of cancer. This is clearly not normal because otherwise this would have been relevant for most other vaccines. But this was a new issue. And what we had found was up to 2 to 3% of the population of that cohort, even after 25 days, still had high activation of their lymph nodes. I'm realizing now that this fits with these epigenetic changes that are occurring within macrophages. And guess what? Even more concerning is that condition that we call long vax. Now, some people still don't believe it exists. So before I say anything, it's important to note long COVID is real, meaning that there are people who have just the infection, they're unvaccinated, and they can have long-term immune impacts with regards to just the infection. That's real. That was known even before vaccines rolled out. Now, some people think that what we call long COVID is actually all vaccine related. Again, I will say that's not what the science says. However, we do know that there are a number of people who, after they have been vaccinated, end up with symptoms that are almost exact to what would happen after the infection. And then we don't know what happens in terms of the risk when you combine the two. Now, they would argue, some research says that some vaccines are protective, others say that some people will get worsening symptoms. It's all quite mixed up. However, this issue with regards to epigenetic changes in macrophages suddenly makes a lot of sense. If you have an immune system that is already partially activated, and then you have either infection or the mRNA vaccine doing epigenetic changes on the DNA, so opening up areas of the macrophage, this is an immune cell, a very versatile immune cell, to then produce cytokines, it is no surprise that it can't switch off. This is pretty serious stuff, and it's not easy to try and figure out how to fix it. Now, just in case people had come late, as I can see that there are a number of people here, in closing, I want to show you again the principles as to what we're looking at. So I'm going to take you back through it very quickly so that you understand the science. Essentially, the paper has found that the mRNA vaccines can reprogram changes in the way how the DNA is able to be transcribed. So it doesn't change the DNA, it just has changes with regards to histones. Now, to understand what that looks like, here are the steps. First, this is your chromosome. You have 23 of them, 23 pairs. If you break down a small piece of it, this is what it looks like. It's very tightly wrapped chromatin, looped around. If you take a small bit of that, this is what that looks like. These loops are right there, but they're still very tightly packed because this is what they look like. And the DNA is wrapped around these histones like a yo-yo, and they're all connected closely together. And if you look even more closely, you will see this yo-yo, the DNA is wrapped around it. Then it goes to the next one, wraps around it, next one wraps around it. This here is your strand of DNA. So the principle is You can't transcribe any DNA when it's all locked up like this. You have to open it out. And what it seems that happens with regards to mRNA vaccines is that they open out the histones or the DNA so that cytokines can then be transcribed. And this is what it would look like. Here you have condensed chromatin in the chromosome. You can't read this. It's too tightly packed together. And this is how this works. This process keeps it very tightly packed so that only the DNA that you want to write is then opened up. So in order for the DNA to be written, you need this process to happen where you unloop from the histones a piece of DNA, which can then be transcribed. This is the process that it seems as though is happening in macrophages after mRNA vaccination in parts of the chromosome or in parts of the cell that produce inflammatory cytokines. As I said, they think that's a good thing. I'm not too sure, because if it goes on and doesn't go off, because these histones are like a dimmer switch, they are able to turn up the light or turn it down. What happens if they are turned up and locked in the on position? You will never go to sleep, and neither will that immune cell. So these are all very complex topics, fascinating topics, and I've just taken you on part of the journey with me as to what I think is going on. There's still a lot more work to do to try and figure it all out. But yes, if you want to continue with joining me in that journey, remember, click on the description, in the description on the links below. Let's see if we can continue this journey otherwise. Have a great evening.